Hi, I'm Kolby Jardine, and I am a staff scientist in Berkeley Laboratory in California, spending 1 year here at Volt on a sabbatical research project studying plant and microbial C1 metabolism. And we have the rare chance to import some specialized instrumentation to study those processes in plants. So I've taken quite a long path to get to the research area that I'm working in now, which is quite interdisciplinary. I've always been fascinated by chemistry, and I first started out as a undergraduate looking at chemical engineering. And then I started to get into more of the biology, and I was very fascinated about the idea of some of the chemical processes that I was studying applying to cells. And so I transferred to a different school in New York and majored in biochemistry and kind of took a long path from there as well. I started to get more into nanotechnology, sort of this artificial engineering of molecules. And I then went to graduate school to do some artificial photosynthesis of creating sort of these artificial systems that could capture the sunlight and convert that into stored organic carbon. And then, but that was strictly sort of laboratory based, and I have a very strong love for the outdoors and outdoor sports, and so I eventually decided to go to graduate school to study ecosystem processes and atmospheric science and terrestrial ecology. So some of the main questions of my research involve the metabolism of plants and volatile organic compounds. Historically, they've been studied in terms of atmospheric chemistry and their impact on climate, where I've, over the last couple of decades, have looked into their roles in plant physiology and biochemistry and how we can sort of use the exchange of some of these gases with plants to understand certain biochemical and physiological processes that are very difficult to study otherwise, especially non-destructively in real time. And when I was in Brazil and California doing field and lab work, I discovered two really potentially very important biochemical processes in plants. One is called the oxidative C1 pathway, where plants take simple C1 carbon and oxidize it all the way to CO2. And then more recently, I discovered a photosynthetic C1 pathway in which plants take CO2 in the atmosphere and convert that into C1 compounds or related compounds in the plant. So the importance of C1 metabolism in plants is pretty well established. It's involved in the transfer of single carbon atoms in the form of methyl groups to biomolecules like DNA and RNA, proteins and lipids, and small metabolites. And that's known to be critical for their biosynthesis and the production of biomass, but also the regulation of the biological function, such as when the cell walls start to grow, for example, they start to release methanol from the cell wall in a tightly coupled process. And when DNA gets methylated, it changes its gene expression behavior, which is critical for responding to environmental and developmental cues. And so we know that it's a central component of plant development and growth, but also stress responses, for example, high temperature and drought responses, in which the C1 metabolic network becomes really important in being able to acclimate to these stresses. A lot of the uncertainty comes in as to what are the sources of the C1 carbon. And I sort of think about it as a soccer game where you have all these single carbon soccer balls getting tagged on to different substances by different soccer players. and we don't really know what the flux of those soccer balls is and what the source of those soccer balls are. And one of the things that we're looking into in Volt is the role of photosynthesis in providing those soccer balls to the C1 metabolic network in a new pathway that we have proposed called C1 photosynthesis. And interestingly enough, that pathway involves the assimilation of sulfur and nitrogen as well, and to form the critical nutrients into the organic form, such as amino acids. And so we don't know how those are coupled and related together. Another major uncertainty of C1 metabolism is the influence of atmospheric volatile organic compounds that only have one carbon, like the methanol, methane, and formaldehyde, in which they may come into the plant and be oxidized all the way to CO2 and integrating into that C1 network. Some other major uncertainties that we have are how the C1 metabolism of leaves are undergoing photosynthesis is coupled with the C1 metabolism of stems and roots that are not doing photosynthesis. And so how are these C1 intermediates transported, for example, in the phloem and the xylem? So one of the reasons why there's not much known about C1 metabolism is that some of their intermediates are volatile gases, and they are emitted to the atmosphere or then consumed from the atmosphere. And most metabolomic studies of plants miss those gases. And at Volt, we specialize in trace gas exchange between the atmosphere and plants and soils. And so we have very high sensitivity instruments based on mass spectrometry and optical spectrometry that are high precision, that can detect very small changes in concentrations or fluxes of very specific C1 gases, together with the classic CO2 and water gas exchange when you're doing photosynthesis and transpiration measurements of plants. And so by coupling those instruments to different scales, for example, at the entire branch scale, we can develop dynamic gas exchange systems of branches. Also, leaf level, where we can look at individual leaves under controlled environmental conditions of light and temperature, but also looking at other types of manipulative experiments, such as what we're doing here in the greenhouse with poplar, where we are manipulating the nutrient supply to the trees in the root solution. And so we are also employing some very novel isotopic studies where we're giving the plants C13 labeled forms of the C1 gases, both as carbon dioxide to probe the photosynthetic input, but also in the form of C13 labeled methanol and formaldehyde and other C1 gases to probe the entry of those gases into the C1 network, where we can now study that by providing them in controlled concentrations continuously and measure the uptake flux of those and measure the downstream metabolic processes by tracing that C13 throughout the C1 network. And so we conduct these experiments for a period of time, sometimes for a couple of hours, sometimes an entire month. And then we flash freeze the tissue in liquid nitrogen and try to find where that C13 carbon went in the different biomolecules. Okay, so the two different experiments that we're working on now with the oak plants and the poplar plants are looking at different inputs of the C1 metabolic network. For the oak plants, one of the cool things that we are finding is that the gaseous formaldehyde that we provide the plants are very efficiently consumed by the plant. No matter what concentration, the more that we give the plant, the more that it eats or takes up. And we don't see, even at quite high levels of formaldehyde, any toxic effects on the plant physiology in terms of the stomatal conductance or the photosynthetic capacity, suggesting that formaldehyde is an entry point into the plant C1 metabolic network, and it's highly efficient at removing it from the atmosphere. And formaldehyde is really interesting because it is the main, it's all the different VOCs, both biogenic and anthropogenic in the atmosphere, during their oxidation process, they pass through formaldehyde. And so formaldehyde is known as a sort of a tracer of these atmospheric secondary chemical reactions. And so by showing that the plants are able to very efficiently assimilate that formaldehyde is quite interesting. And the bigger question that we're trying to answer now is like, well, what are they doing with that formaldehyde? If it's not a toxic sort of side reaction, if they're actually funneling it into the C1 metabolic network, we need to use the C13 labeling to really look at those details. And on the poplar experiment with the nutrients, well, one of the things that we're just testing right now is if we can remove the sulfur and nitrogen nutrients from the poplar solutions, from their roots, is there any impact on their growth or their photosynthetic processes in the leaves? And it looks like the answer is yes. There's quite a dramatic impact, negative impact of removing those nutrients from the roots. And so we are trying to quantify that and also try to eventually try to recover those stress responses by providing those nutrients back, but in a different form in a gas, in the gas phase, instead of as the typical nitrate and sulfate that the roots normally get from soils and so forth. So this research connects to broader applications in agriculture. For example, trying to understand how plants can grow faster and with limited nutrients and water. For example, the C1 metabolic pathway may be important for plant growth, and so that could be selected for by traditional means or genetic engineering means to increase productivity and nutrient assimilation. There's also some evolutionary implications of this research to understand where photosynthesis came from, oxygenic photosynthesis, how it evolved, and how is it connected to growth and the C1 metabolic network. And so, yeah, both in terms of the evolution of plant productivity, but also the future utilization and engineering of this pathway. So the thing that I enjoy the most of this research is integrating fundamental plant biology measurements and science with technical engineering, such as the development of new trace gas sensors and automated workflows and custom branch chambers and looking at across different spatial and temporal scales, like from individual leaves up into entire plants and even entire ecosystems, looking at very fast processes across a few seconds to seasonal and even decadal processes at the larger temporal scales. And so, yes, so I think the integration of science and engineering is what really makes me excited about this research. Okay, thanks for watching, and feel free to reach out and contact me or anyone else at Volt for further information or collaborations that might be of interest. And make sure to watch other videos from Volt. Thank you.